mission is really to take cells and make them into products. We would like to give solution for a range of industry, really industries from like agriculture, food tech, and of course the pharma, which is what we're going to talk about here today. We've been working with cells over the past 2 decades. We really know how to isolate the cells. We know how to manipulate the cells and make them do things that we like them to do ex vivo. We know how to expand the cells and really allow us to produce high quality and quantity of cells in mass scales to fulfill the industry needs, the industry that we talked about. So we have cells from different sources. We have cells from plants, we have cells from animals, and of course from human tissue, which is what the pharma vertical is focusing on. And most extensively with respect to human tissue, we've been using human placenta. So Pluri, really, if you think about it, we like to say we are a placenta company. We're working with placenta for many, many years. The placenta is a very good source of many, many potent cells. There's no ethical issues when we work with the placenta, right? The placenta is actually a medical waste. It's an unlimited source, and we actually been working with local hospitals around here to receive placentas on demand and use them as a source for many, many cells for the pharma. Currently, we are actually isolating immune cells from the placenta, and we announced the launch of a new immunotherapy platform using immune cells from the placenta that called MAIT, mucosal-associated invariant T cells, as an allogeneic treatment for solid cancers. When you think of MAIT cells, you think of a very, very special immune cells. So if you think of the immune system is usually divided to two. There is the very specialized adaptive immunity and the very general innate immunity. And MAIT cells are kind of sit in between. So it have features of both the adaptive and the innate immunity. So MAIT cells are definitely part of the adaptive system. It's part of the T cell population, and we usually actually call them unconventional T cells. But they also have some very special features of innate cells on the other hand. So they can act very, very fast. They can be a first line of defense against pathogens. So we have here a cell that is both specialized adaptive cell and really a fast-acting innate cells. But MAIT cells are not a regular T cell, right? We say they are unconventional T cells. And I think that the major difference between T and MAIT cell, especially when you talk about an allogenic setting, is when one uses T cell as an allogenic treatment, one of the major concerns in the industry is the development of graft versus host disease, or GVHD. Now, MAIT cells are very, very unlikely to cause GVHD, and this is because they have a very unique TCR, the T cell receptor, unlike the T cells. So the T cell TCR is recognizing a peptide antigen that is loaded on MHC molecule. MAIT cells, on the other hand, recognize vitamin B metabolite that is made by bacteria, so there is no reason for it to cause a GVHD, which is very, very attractive in the setting of allogenic treatment. Now, with respect specifically to solid tumor, there are many more advantages to MAIT cells compared to regular T cells, and I think one of the main challenges, one of many, but one of the main challenges in solid tumor treatments is the migration of the T cells into the tumor, or the insufficient migration of T cells into the tumor. Now, MAIT cells are the perfect immune cells in that respect, because unlike T cells that are found in blood, MAIT cells are mainly found in tissue, and this is actually where the name comes from, mucosal-associated invariant T cells. Secondly, they have a very, very unique high expressions of what we call chemokine receptors. Chemokines are those proteins that are responsible for the movement of immune cells in the body, so they can guide the migration of the MAIT cells into the tumor. So if I think of the most important points about MAIT cells with respect to an allogenic treatment, and specifically for solid cancer, they are specialized, fast-acting cells, those cells that lie in between the adaptive and innate immunity. They are very, very unlikely to cause GVHD, making them a perfect source for allogenic treatment. And they have a very high potential that lie really in their biology to migrate into solid tumors. We know that MAIT cells have a huge potential in immunotherapy, and specifically for solid tumor, like we said, but working with these cells can be really can be challenging, and this is from experience. So first of all, if we talk about number-wise, MAIT cells frequencies in the blood are fairly low, and the variation between individuals, if you're looking at them, is very, very high. I mean, we have anywhere between 0.1 to 10% of the entire T cell population to be MAIT cells, so that's very low. The main site of MAIT cells is really the tissue, and some tissues you can find up to 40% of the T cells to be MAIT cells, like the liver, for example. So if we are isolating the cells from the blood, you already start with a very low number of cells. So expanding into mass scale is challenging. Now, if you start with the tissue, that's already a different issue, right? And we are starting with the placenta. And very interestingly, the placenta, which is our source of cell, is highly enriched in MAIT cells. So we're already starting with a high number of MAIT cells compared to peripheral blood, which always helps. Now, in addition, the expansion ex vivo is also very, very challenging, both with respect to the quantity and with respect to the quality. It's very hard to maintain the functions of the cells and the characteristic following expansion. And Pluri, over the past few years, developed a really good 3D expansion technology. This expansion technology allows a more physiological growing condition than the regular 2D platform. So we were able really to expand the cell, receive high quality and quantity of cells, while really maintaining their characteristics and their functioning. If you think of immunotherapy in general, it has been showing very, very promising results for the past few years. And specifically, if you think of CAR T with respect to hematological malignancy, we can see a very, very high response rate with more like 6 autologous products that are approved over the past few years. However, liquid tumors are only about 10% of the entire cancer cases. 90% of the cancers are actually solid tumors. And this really nice efficacy that we see with CAR T for liquid tumors does not translate to solid tumors. So there is a really high unmet need here. And this is for many reasons. I mean, finding the right antigen, the tumor microenvironment that is very, very suppressive, infiltration, migrations of cells that is insufficient, persistence of the cells that are not staying too long in the body, and by the time they get to the solid tumors, they get exhausted. Now, we believe that MAIT cells have a huge potential in overcoming at least some of those challenges. Like we said, they are very, very specialized, fast-acting potent cells. They have intrinsic ability to migrate into solid tumors. They are very unlikely to cause GVHD as opposed to the regular T cells. And all of these features that are lying in their biology, combining with our technology of 3D expansion, really allow the expansion of mass scales in high quality and high quantity that we believe can overcome the problem and really become the next solution for, this devastating disease.